Scientists, for the first time, just grew the first ever mini brains from multiple cells of people. Hybrid brain organoids could be used to test how people respond differently to drugs before clinical trials begin, researchers say. And you're going to be shocked. These are the images of chimeroids. They're grown from stem cells from multiple people and contain an array of cell types found in fetal brains. So they're the type of cells found in the brain of conceived babies. Let's put it this way. So for the first time, scientists have grown 3D brain models using cells from multiple people. The new hybrid creations, which researchers have dubbed chimeroids, are a variation of brain organoids, tiny 3D models made of tissues that mimic the structure and function of a full-size brain. These models are more accurate to human biology than 2D cellular models or animals like lab mice. Because of this, scientists hope that the models will accelerate drug research and development. Typically, brain organoids are grown from cells that are collected from just one donor, and this means that they cannot capture the genetic variability that exists between people, which can impact individuals' brain development and their responses to drugs. Creating chimeroids could overcome this hurdle, according to the scientists behind a new study published June 26 in the journal Nature. Such a village in a dish, quote unquote, could be particularly useful in the early stages of drug testing, they said. Researchers have previously grown sheets of brain cells from the stem cells of different people, but this is the first time ever 3D models of the brain have been grown this way. Chimeroids are an exciting tool that will be widely adopted in the field of neurodevelopment, probably with diverse applications. Aparna Baduri, assistant professor of biological chemistry, University of California, Los Angeles, who is not involved in the research, said. And to make the chimeroids, researchers collected stem cells from 5 people and then in the lab used growing indicating chemicals to coax them into growing into brain organoids, each of which had cells from just one person. The scientists then tore the resulting organoids apart and recombined the cells within them to form chimeroids, and this ensured that each chimeroid contains an equal number of cells derived from each person. After 3 months, the chimeroids were about 3 to 5 millimeters, that's about 0.2 inches in diameter, and contained all the same types of cells that are normally found within the cortex, the outermost layer of the brain of a fetus. A human fetus, yes. Separately, the team exposed the chimeroids to 2 neurotoxic chemicals, ethanol, which is associated with fetal alcohol spectrum disorder, and the anti-epileptic drug valproic acid, which can increase the risk of birth defects. The team found that cells that came from different donors responded differently to these drugs in terms of how extensively the chemicals hindered their growth, for example. If scaled up to contain cells from even more people, chimeroids could theoretically help determine how patients will respond to drugs before they are tested in a chemical trial, the team said, and they could then be segregated into specific treatment response groups. I'm excited about what the future holds in terms of using organoids, such as chimeroids, to develop brand new ways to develop therapeutic innovation for neurological disease. Paula Arlota, co-senior study author and professor of stem cell and regenerative biology, Harvard University, said. And this is by Emily Cook on Live Science. Please leave your comments and thank you for your support. Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.